Uh, Welcome, everybody. I'm Debashis Mitra, the host today of the Data Science Institute's Data for Good webinar. Our speaker today is Dimitris Anastassiou. He's professor of electrical engineering, and he's representing the Sense, Collect, and Move Data Center within DSI. The title of his talk is Biomolecular Data Analysis using the Attractor algorithm for the discovery of biological mechanisms in cancer. A quick word about Dimitris. He received his PhD in electrical engineering and computer science from Berkeley in 1979, was at IBM Research at Yorktown Heights for 5 years, and he joined Columbia's electrical engineering faculty in 1980. He has been director of the Genomics Information Lab at Columbia since 2002. And we will be hearing about this work today. But I want to spend a minute to talk about Dimitris's previous life. He was Associate Director of Multimedia Applications in the Center for Telecommunications Research, CTR as we knew it, at Columbia from 1993 to 1996. Multimedia Applications is smack in the middle of the Sense, Collect, and Move data space. Back in the 1980s, the internet was largely just a vision. Packet technology was just starting, and packet voice was more or less the only app. Dimitris's work laid the foundation for video technology. His patents are incorporated in the MPEG-2 standards, and is truly a pioneer in the video technology and, in fact, the video industry as we know it today. That's all I wanted to say about Dimitris. A quick word about the webinar protocol. Unfortunately, participants cannot speak. Only the host can, but but participants can type their questions in the chat section during the lecture and later, and Dimitris will read out the questions and answer the questions after the lecture. Okay? So Dimitris, it's all yours now.
Thank you so much for this great introduction. I'm delighted to give this seminar. So yeah, it will be, I will present results that happened in what I call after I finished my previous lifetime, which you summarized. Yeah, in the year 2000 approximately, I changed my field and I became a born-again molecular biologist. Okay, so let me share my screen. I have to share the PowerPoint. And okay, so is everything in order right now? You can see the title screen, full screen? Yes, excellent. Okay, yeah, so the general topic will be consistent with the theme of this series, how we can collect data from, particularly from cancer biopsies, biomolecular data, which we can analyze for good purpose. In this case, they are cancer data, and the purpose obviously is to shed light on the mechanisms that would hopefully lead to therapeutics. So I will start because not everybody in the audience is familiar with molecular biology, talking about some essential concepts. And then I will go to a particular example about which we are very excited about. We believe that there has been a discovery of of an important biological mechanism in cancer, which is associated with invasiveness, metastasis, and resistance to therapy, and that this was discovered purely by data analysis. And that will be the topic of the second half of the presentation after I discuss about all these prerequisite concepts. So in a nutshell, there will be these topics. I will start first by describing a particular cell population whose gene signature we discovered about 10 years ago, and that is really what is associated with invasiveness and metastasis. This is what we call the COL11A1 expressing cancer-associated fibroblasts. Then I will describe the particular computational methodology that we use, which is non-traditional for this particular application, which led to the conclusion that there is a cellular transition. Some cells transform themselves, they differentiate, and the origin of this transition is a population of cells that reside naturally in normal fatty tissue, which is called adipose tissue. And we call those adipose-derived stromal cells, or ASCs. And so we believe there is a transition from ASCs to the COL11A1 expressing cancer-associated fibroblasts. And the underlying vision is that we need to find potential therapeutic targets that would inhibit this transition. And that would be wonderful if it could be achieved, because it could be applied to multiple types of cancer that would inhibit this mechanism and would hopefully inhibit the imminent metastasis. So first, I start with some essential concepts from molecular biology. So we have numerous cells in our body. Each of them has the same genome in DNA. You can think of it of a sequence of letters, A, C, G, and T. And this sequence contains segments which define genes. And in a simplified manner, we can say that we have approximately 30,000 genes, and each of these genes can be expressed, meaning that under some conditions, they create their own, they generate their own product. And the product is the protein, RNA and then protein. But in this case, we don't need to know any details. We just can say we have product biomolecules. And so each gene expresses some of those molecules and in various quantities, and these molecules essentially define the nature of the cell. And numerically, the expression level of each of these genes determines the amount of the products that it generates. And there are many cell types. We have the brain cells that are called the tumors, the immune cells, etc. Each of those cell types is different from another. They all have the same genome, but they are different, and they are characterized by a particular gene expression pattern. And so that's the main concept that I will be dealing with. We can measure the gene expression level for each gene and actually each individual cell. And so consistent with the theme of this seminar series, we do, the topic of sense, collect, and move, we're dealing with the biomolecular data, and particularly from cancer biopsies. And thankfully, these data become increasingly available, publicly available. So there are extremely rich data sets currently, which are ready for data analysis, and I believe that that means that there are biological discoveries waiting to be made. Now, the previous technologies could measure the expression level, the aggregate expression level for every cancer biopsy, which means that it would correspond to a mixture of numerous cells of different types This is called bulk gene expression data. And so when we have in a particular biopsy sample of a patient, when we say this gene has this expression level, we are referring to the aggregate expression level by essentially averaging all the various cell types, because some cell types would express it, some would not. And so it is much more difficult to make sense out of that. It can still be very helpful, as I will explain now in the next slide. However, what happened with the recent technologies in which we can go at higher resolution at the single cell level is, in my opinion, a paradigm shift, because now computational data analysis, when applied on those data sets, if they are particularly rich, this may be sufficient for the discovery of biological mechanisms even without experimental verification, just by data analysis alone, in my opinion. And so in cancer in particular, the cells which become malignant are those that have some modifications which make them divide abnormally and divide uncontrollably and create tumors. And that's one part of the story, that there's a growing tumor. But the worst news is that these cells can become invasive and they can metastasize. And this happens as a result of their interaction with what is called the tumor microenvironment, which contains many other types of cells, which are normal. They are not malignant, but somehow the tumor cells have learned to interact with them in various ways and use it for their survival, their benefit, and they learn to become invasive and move to other issues. This is often accompanied by the generation of some particular types of cells, which are called cancer-associated fibroblasts, or CAFs. And I will be referring to those particular types of cells in this presentation. And this happens again in the tumor microenvironment as a result of their interaction with cancer cells. And those cells, the cancer-associated fibroblasts, have different expression patterns. There are also several types of those cells. And I will start my presentation talking about specifics by referring to a particular type of cancer-associated fibroblasts whose gene expression signature we actually discovered about 10 years ago. And the actual final contribution of the research I will describe is that it actually sheds light on an important, well-known role that fat is playing in cancer progression. So the fatty tissue is called adipose tissue, and there have been numerous papers. So here is a list of 5 of them in journals, prestigious journals that have been published recently, and they all confirm the well-known fact that the adipose tissue contributes to cancer progression in ways that are not well known. So this will be a result that will shed light on the mechanisms underlying this fact. As I said before, using traditional bulk technology, we can measure the expression level of genes in many samples. Every sample is a patient in that case. And so we can create a gene expression matrix containing the gene expression levels. And the rows of this matrix correspond to the genes, as I said, 25,000-30,000 genes. And the columns represent patients. Every patient has one sample. And so that has been the way that we have been working in the past. And recently, in the recent few years, as I said, we have single cell technology, and that changes things because the gene expression matrix, the columns of the gene expression matrix, rather than being samples or patients, they are now individual cells, and this is remarkable. And that allows us to really understand things in much more detail about what happens inside the cell. So I will start presenting, as I said, some results that we had about 10 years ago. I will show a gene expression matrix. As I said, the rows are genes and columns are patients. And as we know, there are computational techniques like hierarchical clustering. So we can use hierarchical clustering to rearrange the rows and rearrange the columns so mutually associated vectors become close to each other. And this can be visually represented in what is called the heat map, where high values are red and low values are blue. And so 10 years ago, actually, and this is something that reminds me of that time, the first experiment that led to this observation was that a student of mine created this heat map from an ovarian cancer data set in 2010. Again, rows are genes and columns are patients. And we observed this red square, and where my cursor is right now shows the IDs of actually 41 samples, 41 patients, in which a particular set of genes was overexpressed only. So that actually, I mean, without any doubt, represents something. So the question was, and as if we magnify on the right where there are the gene names of every row, if we focus on the heart of this red square and we magnify it, we are going to see many gene names. And I will mention 3 of them because that is really what we discovered to be at the heart of this mechanism at the time: collagen, col11A1, THBS2I, and HPA. We don't need to know what these things mean. We can just take for granted that these are 3 important genes that were found by this experiment. So the question that I asked my students at that time is, what do these 41 patients have in common? Can we look at the phenotypes in the data set to understand if they have something in common? And that was not very easy because there are all kinds of these phenotypes: the age of the patients, the grade, the stage, etc. And in the end, we found it. The answer is that not even one of those 41 patients was in stage 1 or stage 2 cancer, ovarian cancer. They all were in at least stage 3. And this is something that cannot be due to chance. So that means that this signature is generated only when ovarian cancer reaches stage 3, not in all samples that we had, but it never happened before that. That means something could trigger it at that time. And then we spent several more days because it was intriguing that we found precisely the same signature in various ways with prominent presence, again, of those 3 genes that I mentioned in other cancer types, like in colon cancer or in breast cancer. And now that we knew the stage association, we tried to say, is it really true that only stage 3 colon cancer patients have it? No, the answer is that stage 2 also had it, but not stage 1. And in breast cancer, actually, it was more tricky because they had it even from stage 1, but it turns out that they didn't have it before that. There is a condition called DCIS, in situ, for example, and that never had it. So with differential analysis of genes between cancer samples below this threshold and above, we could find this signature very clearly. and then it was clear that it represented some kind of an important mechanism, because the presence of the signature, as I will explain, was associated with bad news for the cancer patients. And a potential explanation for this stage association is that, and it was also clear that this signature was expressed by cells that were fibroblasts, because the nature of all the genes that were overexpressed suggested without any doubt that these were fibroblasts. And so naturally they would be cancer-associated fibroblasts or CAFs. And the explanation was that they happened when tumor encounters adipose tissue, fatty tissue, because in ovarian cancer, stage 3, only in stage 3, cancer cells can reach the omentum, which is a fatty tissue. In breast cancer, fatty tissue exists from the very beginning, except for in situ. And in colon cancer, there is not adipose tissue in the beginning, but when it goes to stage 2, it has already spread to have meta-adipose tissue. So this is what happened at that time. And then we also found that we don't really need to do differential expression between stages to find the signature. We tried to rank all genes in terms of their association, and the association measure that I like to use is called mutual information. We found that by ranking genes in those data sets in terms of their association with gene COL11A1, it consistently and accurately identifies the signature. And it is the only gene that has this property. In other words, if we start from THBS2 or INHBA and we rank all the genes in terms of their association, then we would not find such an accurate version of the signature that we already knew. And this is consistent with the fact that, as I will explain, this is really the final endpoint of the transition that I want to describe. So a few more slides just to give the final convincing arguments that this is for real, is that if we just look in supplementary tables of from very old papers, even before 2010. These are from supplementary tables from papers in 2006, 2007, and 2008 from 3 different cancer types: breast, ovarian, and pancreatic. And the authors at that time had not realized that there was anything interesting in these rankings. They just presented them. But when we look at them now, we see, they ranked in terms of the so-called fold change. In breast, it was between, as I said before, between in situ, which is stage 0, and an invasive ductal carcinoma in this case. In ovarian cancer, actually, it was between omental metastasis and primary tumor, and omental metastasis is stage 3. In pancreatic, also there is a peripancreatic fatty tissue very close. And so we really, this cannot be due to chance. If we find the genes that appear in all 3 tables, or in at least 2 of the 3, they are color-coded green or yellow, the p-value is ridiculously small, 10 to the minus 100, something like that. Okay, so the signature is there. And since then, during the last 10 years, there have been numerous papers from authors who had independently, in most cases without even being aware of what I have just been describing, they kept discovering that these genes, COL11A1, INHBA, THBS2, they are all important biomarkers, or they are associated with the resistance to therapy in various cancer types. So the first one is lung, the second is pancreatic, the third is ovarian, the 4th is prostate, the 5th is esophageal, then gastric, and then the other genes again in the same thing. So, okay, that's enough for the first presentation. We have a signature, and I'm going to claim this is the final endpoint of a cellular transition, which is definitely, as we see here, associated with all these bad things that we would like to inhibit. And also the exciting thing is that this is a pan-cancer phenomenon. It happens to all cancers. So if there is a therapy that would inhibit this mechanism, that obviously would be huge. Okay, so this is, again, what I just said. This is a conclusion that this is a characteristic signature with prominent presence of 3 particular genes. And so, yes, so again, 10 years ago, we made this computational discovery. It has not been noticed, and the signature has not been appreciated to the extent that it should have, in my opinion. But anyway, now we have the opportunity to really understand what it is and do something about it. So the next step is to find out how these cancer-associated fibroblasts that express those genes, how are they generated? And now we do the data analysis on the single cells. So next topic I'm describing is the Attractor algorithm. Which is something that we actually presented in 2013. And actually, it is a very intuitive, very simple concept. It is a data analysis algorithm that can be applied in all data sets where you have many features and there is a vector for any feature. So in this case, the feature is the gene. And we have a gene expression matrix. And as I said, we can evaluate the association. I like mutual information as an association measure because in my previous lifetime, I was an information theorist, and I like this measure. It can capture nonlinear effects. It is much more appropriate than, let's say, the Pearson correlation, in my opinion. And so we can rank all genes in terms of their association with a particular gene. So I will start going back to the bulk gene expression analysis, just giving an example that clarifies what the algorithm is. It is iterative. It is an unsupervised methodology, like clustering, but it is not clustering into mutually exclusive segments. It is something else. It starts from a seed gene, and essentially it keeps iterating. If there are many other genes that are very highly associated with it, a group of genes, it takes them together, it creates a fictitious metagene, which has the average, something like the average or a weighted average of the gene expression of those genes, and it keeps re-ranking those genes. So we every iteration ranks genes in terms of a metagene from the previous iteration, representing the consensus of co-expression. So that's the, I will illustrate the concept. From an ovarian cancer data set back in 2013, this is from a slide that I used in the presentation at that time. Let's take one gene. We don't need to know what it is, but its name is MMP11. We rank all the genes in terms of their mutual information with MMP11 at iteration 0, and we get this ranked list of genes. Now, I will simplify the algorithm. I will say, let's say that we take all those genes that we see in this first column, and we think of the metagene, which contains the average expression of all those genes. Now, using this as seed, let's re-rank all the individual genes in terms of their mutual information with that metagene. And this is the result, iteration 1. It happened that MMP11 remained at the top. Usually every iteration is similar to the previous one. But here we see significant changes, call 5A2 moved up, etc. So we have a new ranking. Now, next iteration, we consider a new metagene which has an expression level for every gene, which is the average among all, it's the average among all those genes, I'm sorry. And then we rank all the genes in terms of that metagene. We get a new ranking. Now things have changed significantly. MMP11 went all the way down in this column. Next iteration, actually, it disappeared, and remarkably, it looks almost identical with the previous one. In iteration 10, it changed a little bit more, and then it doesn't change at all. So obviously, it converges. And so very quickly, I want to say that this is called an attractor. The seed gene MMP11 from which we start can be thought of as the attractee gene. And the concept is that starting from any of the genes that we see in this slide, we will converge to precisely the same attractor metagene. So for example, GRAM1, I don't know if it exists at the bottom of the second column. So if we start from GRAM1 as the seed gene, I will demonstrate how we will converge to precisely the same attractor. So I will compare it with what I already described, starting from MMP11 at iteration 0, and now I put the association measure values, the mutual information. So naturally, it is a normalized measure as we do it, and it starts from exactly 1 in both cases. So ranking all genes in terms of MMP1 gives this ranking, which is what I had showed before, the first column. And ranking genes in terms of GRAM1 gives something different. We see there are many common genes, which is consistent with the fact that they are related, but they're not the same at all. So now I'm moving to iteration 1. Iteration 1, the 2 seed genes remain at the top, but their weights now are not 1 naturally, because now we rank in terms of their mutual information with the previous metagene, not with itself. So it went down a little. And the ranking has slightly changed. Okay, now we go to iteration 2. So already, as we saw before, FAP, fibroblast activation protein, is ranked number 1. And even everything else seems to be identical in the 2 rankings, not exactly, but almost. And the numbers even are getting close. In iteration 3, extremely quickly, that becomes even more prominent. Iteration 10, with the rankings are exactly identical, even with 3 decimal digits. I think there is only one difference. This 811 is 810 here. Everything else is identical. And in iteration 20, even this has become. So without any doubt, the algorithm converges, and mathematically one can, it's not very easy, but one can deal with these things and prove conditions for convergence. So to make the long story short, this algorithm from numerous attractee initial genes, we converge to precisely the same attractor metagene, which represents a co-expression signature. And there are many such co-expression signatures, each of which has its own set of many attractees. And so if we do an exhaustive search starting from all the genes, or a heuristic exhaustive search if we want to minimize the complexity, and if, let's say, we only accept as a valid attractor signature one in which the strength of the 10th gene, where strength can be the weight of the 10th gene, like before, this is almost the attractor. You see all these numbers are very high. If it is more than 0.5, then it means that it is very, very strongly associated. So if we put as a condition that the 10th ranked gene has some kind of a large, more than 0.5 weight, then in the end, the exhaustive search will yield a limited number, 5, 6, 10, 15, of such attractor signatures, each of which is a co-expression signature that in this case obviously contains at least 10 strongly co-expressed genes, and that represents a biological mechanism because it cannot be due to chance. It represents something waiting to be found, and the beauty of the algorithm is that it converges to the core of the underlying mechanism with this iteration. So the top ranked genes, we end up with a ranking of genes. The top ranked genes can be thought of as being at the heart, at the core of the underlying mechanism, and probably we should pay attention to those. And as I said, this is a general data mining algorithm, does not need to apply to gene expression. It can apply to any data, and it can have any other features as long as we want to find what, if there are many features that are very, very similar to each other, going up and down together, in rich data sets, this is an algorithm that can converge to the core of this mutual association of many features. Okay, so just to say a few words about this, what we did several years ago. So we used bulk technology to analyze data from many cancer types, and we found that many of the attractor metagenes were identical across many cancer types. And this is even more remarkable because it shows that there are some pan-cancer universal mechanisms, and we can shed light on those. So one of those mechanisms, of course, in cancer is that cells divide uncontrollably, and there is a signature called the mitotic signature, and as I said, it was identical. I mean, it is not always the case that in ovarian, prostate data sets, these, we will have exactly this ranking when we apply the algorithm, but applying a consensus version of the attractor algorithm across many data sets leads to an overall attractor, which is very, very similar with each of the individual ones, and they are obviously so close to each other that different data sets, I mean, we don't even know what cancer type we're talking about when we see these results. There is only one universal. And so these genes have to do with mitosis, cells dividing. And so if we take the top 3 genes, TPX3, 2, etc., and we create what is called, and this is associated with tumor grade. The higher is the tumor grade, the more cells tend to divide uncontrollably. And these scatter plot, each of those squares, each of those scatter plots shows all the patients in a particular data set as a dot. And every figure here corresponds to a cancer type. For example, this is head and neck, this is leukemia, this is colon, this is glioblastoma, etc. But the horizontal axis is the first gene in the mitotic signature. The vertical axis is the second ranked gene in the mitotic signature, and we have color-coded the third gene, KIF-C1. And obviously the dots at the upper right of each of those figures correspond to patients with very high grade. And at the lower left, the blue dots correspond to patients with small levels of the mitotic signature, which have low grade. And we see that indeed the co-expression exhibits itself in the form of straight line that goes from blue to red, consistent with the pan-cancer nature of this phenomenon. Okay, and now when we go to single cells, as I said, we have a paradigm shift. Now columns are cells. Resolution is enhanced. And now the attractors are guaranteed to be co-expressed by the same cells, not just by the same samples, which are mixtures of cells. And so we have the opportunity to use a marker for a particular cell type, and the attractor will show us the dominant population of this cell type in the data set. So one commonly used marker for fibroblastic cells, like the cancer-associated fibroblasts, is Lumican, LUM. So using that as seed, we applied that on an extremely rich single-cell data set, happens to be from pancreatic cancer. It contains 57,530 cells from 35 different samples. From 35 different patients, and they are labeled T1 to T24, T standing for tumor. So 24 tumor samples and 11 normal samples from N1 to N11. A treasure of information. As I said, we applied the attractor algorithm in each of them. And so I have rearranged here the tumor samples. And of course, there's no time to go into details, but what we see, I did it on purpose so that the green highlighted gene, which is gene APOD, it's called apolipoprotein 3, and it's an adipose-related gene. We see that it tends, according to this rearrangement, it tends to go down. On the other hand, the red label gene is what I already mentioned, collagen 11A1. It goes in the other direction, and this is true in general for many other adipose-related genes. They go down, and some other genes like THBS2, INHBA keep going like that. And that demonstrates the continuity. That answers the question, how do we know? I mean, as I will say, we have established the final endpoint, and as I will show, the origin as well. And so people say, how do we know that there is not a mixture of these two? This algorithm actually demonstrates visually that there is indeed a continuity. Now, going to the normal pancreatic samples, this is even more impressive. The attractor algorithm, starting from LUM, as I said, LUM, sometimes it's at the top, sometimes it is not. So the attractor algorithm rearranges things, but in the end, it converges to almost precisely the same genes at the top. This cannot be due to chance again. The p-value is infinitesimal. And so when we take the 18 genes, which are shared among the 50 genes in the attractors of at least 10 out of all here there are 11. So in at least 10, we have all those genes, fibroblastic markers like LUM, DCN, as well as adipose-related markers like APOD and DPT, etc. Then when we do what is called gene set enrichment analysis, which is a facility that we give a set, a few number, and a small number of genes, and then it compares it with numerous signatures that have been deposited representing several events. And so the top hit in this case is what is called Boquest stem cell up, coming from a publication by this author who had created a list of genes found to be upregulated in stromal stem cells from adipose tissue versus the non-stem counterparts. So this can only give a hint, and it gave us the hint, yes, it comes from adipose tissue and they have stem-like properties. So we don't need in this presentation to talk about these concepts, but yes, these are stem-like cells naturally present in normal adipose tissue. And to prove that, we said, let's find another single cell data set, this time coming from normal adipose tissue, nothing to do with cancer. So this paper, single cell analysis of human adipose tissue, nothing to do, the data sets that we analyzed had nothing to do with cancer. And in one of the supplementary tables, because bioinformaticians like to use clustering and they take the results for granted. By contrast, we don't do that, because typically the papers that we see, they say, oh, we applied dimensionality reduction with clustering and we found 17 clusters and we give these names, etc. And I think that this is an approximation of biological reality and it can be misleading. So, but anyway, in this case, they found a cluster which they called P5, and the membership of that cluster, found again in the supplementary table of that paper, was remarkably identical to the top genes in the attractor of the normal pancreatic cell. So we see APOD, we see, of course, LUM, we see LUM and PDGS and DCN and all that. Obviously, it is the same thing. And so we applied the consensus attractor algorithm on the normal adipose tissue. Actually, it comes from what is called the stromal vascular fraction. These are not adipocytes. These are not cells, fat cells per se, but they are cells that exist in the adipose tissue. And so the attractor corresponding to this SVF of normal adipose tissue is shown here in the first column. The consensus of the normal pancreatic data sets is there. Again, remarkable similarity. The yellow are those that appear in both lists. And again, we're talking about 30,000 genes. There's no way that by chance all those genes would appear at the top. So that demonstrates that the origin of the transition is a stable normal population existing in fatty tissue. And I say the origin because in this slide, we see LUM and APOD, etc., in some tumor cells already in which cancer has not progressed so much. Okay, so now I go directly to the transition topic. I guess I should go a little faster now. So we claim, I mean, we already have demonstrated now from the attractors that there is a continuous transition between these two cell points. And so this is the paper I would like to give credit to the three students that significantly participated in this research, Kai-Yi Zhu and Ling-Yi Tsai and Chen-Xian Cui. We have a preprint, and now our paper is under review, and we are expecting to hear soon. And so we are claiming that there is this transition of cancer cells as a result, transition of adipose, of cells that exist in the adipose tissue into those cancer-associated fibroblasts as a result of the interaction of cancer cells. So in addition to using traditional methodology, the clustering that I mentioned before, trajectory analysis, etc., we also did what I jokingly call TLC research, standing for tender loving care. These scatter plots, which you can really scrutinize in detail the presence of all types of cells expressing particular combinations. You can see them as dots in scatter plots, etc. We validated that the results in other cancer types. And these are trajectory inference plots. I can very briefly mention that genes like COL11A1 tend to go down naturally. Genes like apolipoprotein 3 tends to go up. And these are several genes that have the properties that I mentioned related to each of those. So these are the genes that are high in the beginning and they become low, and the opposite. And there are some genes that go up and then go down, and this is very remarkable because biologists tell me that they are potential targets for therapeutics. Probably they play roles. If they play driving roles in the transition, then inhibiting them may inhibit the transition. And the continuity of the transition is also demonstrated by scatter plots. On the right, we have the col11A1 horizontal. The vertical axis is, again, the apolipoprotein D gene, which is a marker of the origin of the transformation. And these are scatter plots from two pancreatic cancer patients which have advanced disease, and col11A1 actually exists. So these are definitely cancer-associated fibroblasts. So we see the continuity. It is color-coded for LUM, which is a fibroblastic marker. So these are the fibroblasts, and the transition has gone like that. And these are remnants from the previous, from the origin of the transition. And so I'm almost at the end. Here I show the ranking in terms of mutual information of in 5 of those pancreatic patients. These are tumor samples T23, T11, T6, etc. When we rank them in terms of col11A1, again, we find the genes that I mentioned before in all cases, THPS2I, NHPA, etc. But we also find other genes. One of them is MFAP5. And this is an interesting gene for the reason that I will explain right now. It's actually very often the most ranked gene when we use single cell analysis. And I recently found that there has been some paper by authors from MD Anderson who have discovered that blockade of that particular gene, actually the protein, the product of that gene, MFAP5, enhances chemosensitivity in ovarian and pancreatic cancer, and actually reduces the level of the disease in various ways. So they have developed an immunotherapy, and they're going to start clinical trials related to that. And when I saw that, it was very clear to me that by blocking MFAP5, somehow they blocked the signature, the final signature. And indeed, as I read the paper and I went through the press release, they even say that fibroblasts treated with the antibody showed the lower levels of gene collagen 11A1. So that's it. The gene MFAP5, they say, promotes fibrosis in those 2 cancers, actually in all cancers, and promotes progression, reduces survival of people with those cancers, and blocking this protein is good for therapy. So they developed an antibody that recognizes this protein and it neutralizes it. And they say it also enhances the effectiveness of chemotherapy. Again, I believe this is because it reduces the aggressivity. And this is wonderful. Actually, I feel that this could be a very, very promising therapy. And I hope that it succeeds without significant side effects. And at the end of my talk, I will refer to it one more time, just to repeat that, in my opinion, these non-traditional computational systems biology methods that we're using, combined with having them applied to particularly rich data sets, represent a paradigm shift. And in this case, they led to the discovery of this continuous cellular transition, which is actually part of a more general biological mechanism of cancer invasiveness, which also explains the role of the adipose microenvironment in cancer. And that's my final point. Just like they found that MFAP5 blockade inhibits this final stage of the transition, I believe that all these details about the transition, the genes that go up and then down, who knows, maybe other non-coding RNAs, they're called microRNAs, that may play driver roles. I believe that scrutinizing this transition presents very good opportunities for inhibiting it to be applied in pan-cancer therapeutics. And we don't need to target the final stage. We can target the intermediate stages. There are many potential targets for that, and I'm hopeful that this may happen. So that concludes my talk. Thank you very much. And the next thing for me to do is to stop share, I believe. Yes, and the time is 11:58, and I look at the chat. Is this what I need to do?
Thank you, Dimitris. It's just wonderful, fascinating stuff. Now, a word to the participants. You know, the webinar protocol doesn't allow you to speak, but you can write in your question in the Q&A box, and there's one already there, and Dimitris will answer that. Can you see that one there, Dimitris, in the Q&A box? It's from Tiara Ahmad.
Let me see. I see your Alexis. Hello there.
No, go to the Q&A.
Oh, Q&A.
Right.
I went to chat.
Right.
Are this signature also found in leukemia? Very, very good question. No. In non-solid cancer types, they are not found. So as Tiara can confirm reading the old papers, we particularly refer to solid cancer types. So I believe that it is possible some metastatic blood tumors encountering adipose tissue may have the signature, but it is not happening in the beginning stages in any way. We never saw that. So that's very good. I should have made that clear, actually. So it is good to refer to, when I say pan cancer, I should say pan solid cancer. Yeah.
Anyone else with a question? Type it in. Ah, there's another one.
Sarcoma types. So I'm not a biologist, and I believe that yes, we have found the signature. So is sarcoma related to fatty tissue itself becoming malignant? I'm not sure. No, that's lipoma. The answer is no. We have not seen any differences in the end. Maybe in the very beginning, like before the signature is created, maybe we see hints of the signature, which is not identical, but at some point in all solid cancers, we actually have seen precisely the same signature. So it is totally universal without difference. Because these are, it's not only, rather than referring to the signature, you can refer to the source of the signature, which is totally independent of cancer type, cell type. That's what I believe very strongly from the experience that we had. These cancer-associated fibroblasts are very unique and identical, but I'm not 100% sure if this is totally accurate. Maybe in different tissues it is slightly different. Yeah, but these genes always are expressed like that.
Okay, no other questions? Well, in that case, Dimitris, thank you for this most fascinating talk. It was just a whole world out there that, oh, maybe there's just one comment perhaps from Kausik Raghunath.
So yeah, EMT. This is also a very good question, because 10 years ago, I thought that even the COL11A1 expressing signature was expressed by epithelial cells. And at that time, I was in discussions with Bob Weinberg of the Broad Institute, who is obviously a well-known cancer researcher and one of the most prominent researchers of the epithelial mesenchymal transition, which is the process by which cancer cells themselves become more invasive. They obtain mesenchymal traits. And so sarcoma is a tumor of the mesenchymal cells themselves. And yeah, so the answer is the same I gave before. Even in sarcomas, in the end, somehow the same cancer-associated fibroblasts are expressing col11A1, INHP8, THPS2. We have observed them as well, yes. Yeah, now as for EMT, I'm still curious, but you know, I still am in touch with Bob Weinberg, and about a few weeks ago, he told me that he now believes that tumor cells express less and less of what he thought in the beginning that they express. In other words, he said that it is even possible that they don't even express any collagen at all when they undergo partial EMT. So it is still a mystery. I believe that there are many, many papers that have overemphasized the role of EMT, and I am one of the guilty parties, of course, in that respect. It appears, you know, the more I look at the single cell data and I'm trying to figure out exactly which cells are really tumor cells, it's not very easy to make absolutely sure that they are tumor cells. But the more I believe that these cells really are do not become fibroblastic in the end. They, it is undoubted, they undoubtedly undergo some transformation, but not as drastic as I thought. Yeah.
Well, thank you, Dimitris. Thank you very much.
Thank you very much.
Thank you all.